Welcome. In this session, I'm going to continue discussing pedigree analysis, after which I'll start discussing about genetic disorders. Right. So, in the last session, I discussed about how the inheritance of autosomal dominant trait occurs. In this session, we will see about autosomal recessive trait, so how it shows the inheritance pattern. So before I start discussing about it, I would like to again remind you, okay, what actually is this pedigree analysis. Okay, so this pedigree analysis is a strong tool in human genetics. It's a strong tool in human genetics. I told you that in case of human beings, we cannot make a controlled process that how we have made in other cases, okay, in plants or other animals, right? And the pedigree analysis, okay, in case of humans, it will help in tracing inheritance of a specific trait. So it will help in tracing, okay, inheritance of a specific trait, like okay, pattern, bald head, right, or hitchhiker's thumb, okay, brachydactyly, polydactyly, like this. There are, okay, many traits that we can find out what the pattern they are showing. Okay, then the pedigree analysis also helps in finding out the inheritance of an abnormality. So even we can trace an abnormality, and also help in tracing the inheritance of a disease. So that is the use of pedigree analysis in case of human beings, right? So now let's see about inheritance of autosomal recessive trait. So let's see inheritance of autosomal recessive trait. So I'll take an example for this. It's sickle cell anemia. Okay, so here the recessive traits, the recessive traits always, okay, will get expressed. So the recessive traits always get expressed in homozygous condition. So they always get expressed in homozygous condition. Okay, so say suppose here I take a capital A, so this is the normal condition, and small a is the sickle cell anemia. Okay, so because I have taken small, as I told, it's a the sickle cell anemia is a recessive trait. Okay, so when we see capital A, the condition will be normal. Even in capital A small a, the condition will be normal. So when we see a person inheriting, okay, both recessive alleles from the parents, then it results in sickle cell anemia. So that is what I have mentioned here. The recessive traits are always expressed in homozygous condition only. So let's see here. A normal male marries a normal female, and they have a total of 5 children, right? So first son, the next son, then daughter, then again daughter, and lastly they have again son. Okay, so he's the eldest, so second one, third, fourth, and this is the youngest one. Okay, So total there are five children, and what we have seen, okay, one of the son is showing the presence of sickle cell anemia condition. Okay, so one son and one daughter showing, okay, sickle cell anemia condition, right? So here the parents are heterozygous, because of that they are normal, but in them the sickle cell anemia gene is hiding. Okay, I told you in this condition, in heterozygous condition, okay, in homozygous dominant condition, in these two conditions the persons will be normal. Only in, okay, homozygous recessive condition we will see this sickle cell anemia getting expressed. Okay, now the daughter who seems to be normal marries a normal, okay, man, and they have three children. And in that case the first one is male, second one, okay, a female, that daughter, and third one is also daughter. So one, two, so they have total three children. And in this we see the first child is having the condition sickle cell anemia. Okay, right. So here the parents, so if you take here capital A small a and capital A small a, then we'll see how it's getting, okay, inherited. So the capital A small a, capital A small a, so we have, okay, capital A small a and capital A small a. So this one and this one, capital A, capital A, capital A small a, capital A small a, and small a, small a, right? So there is a 25 percentage of having sickle cell anemia in, okay, in their children. Okay, so this is what we have seen, okay, in one son and in one daughter out of five children. Okay, so here in case of this autosomal linked traits, that is recessive traits. Okay, so we see that the siblings, so here all these are siblings. In siblings are homozygous for defective allele. So here it means these inherited small a and also small a. So others will be either homozygous or in heterozygous condition. Okay, right. So this is the inheritance of autosomal recessive trait, right? So that's all about the pedigree analysis. We'll see the next one, genetic disorders. Right, so these genetic disorders are of two types. They are Mendelian disorders and chromosomal disorders. So the genetic disorders of two types, okay, Mendelian disorders and chromosomal disorders. So here, coming to these Mendelian disorders, they show dominant or recessive inheritance. Okay, so Mendelian disorders will show dominant or recessive pattern of inheritance. Whereas in case of chromosomal disorders, they are not going to show, okay, any dominant or recessive pattern. Okay, now these Mendelian disorders I'm going to classify again into two types, basing on, okay, whether they are of autosomal linked or allosomal linked. So autosomal disorders and allosomal. It means that the genes that are in case of allosomal, the genes are present on sex chromosomes, and if the genes are present on autosomes, then the condition is called autosomal. Okay, and under this autosomal, we will see again, right, dominant or recessive. So dominant or recessive. Okay, so autosomal recessive, autosomal dominant, and similarly we will be seeing in this condition also as allosomal, okay, dominant, allosomal dominant or allosomal recessive. Okay, so here just I'll mention examples. In this case already we have seen the dominant one is myotonic dystrophy. So myotonic dystrophy in case of autosomal dominant trait, right? Then in case of recessive traits, right, I'm going to discuss especially about sickle cell anemia, then phenyl ketonuria, and then thalassemia and cystic fibrosis. Okay, and right, those are the one which I'm going to explain under recessive traits, that is autosomal recessive traits: sickle cell anemia, phenylketonuria, thalassemia, and cystic fibrosis. Okay, and coming to the recessive of allosomal, so under this I'm going to explain about colored blindness. Okay, and also we will see about hemophilia. Okay, so recessive, allosomal recessive, color blindness, and hemophilia. And in this case recessive autosomal, autosomal recessive, sickle cell anemia, phenylketonuria, thalassemia, and cystic fibrosis are some genetic disorders which I will be discussing about. Then coming to chromosomal disorders, here these also I'm going to classify into two types: autosomal and allosomal. Autosomal and allosomal. So if autosomal chromosomes we see aberrations, then we call, okay, autosomal. If autosomes are affected, if sex chromosomes are affected, I say allosomal. Okay, under autosomal I will be discussing about Down's syndrome, and under the allosomal we will discuss about Klinefelter's syndrome and Turner's syndrome. Okay, are some examples that at present I will be discussing. So here, don't worry, this part we will say in objective. So in some cases and many other disorders I did not mention here, so I have taken mostly the examples of the textbook. Later when we see the objective part, I will be telling this again in detail. Okay, so even we will see the examples under this allosomal dominant condition, and not only these disorders which I have mentioned. In addition to this, we will see some other examples also when I discuss the objective part of it, right? So now let's see, starting with the autosomal conditions. I'll start with the sickle cell anemia. I'll see about sickle cell anemia. And this genetic disorder is due to caused due to mutation in hemoglobin beta gene that is located on chromosome number 11. So due to mutation in hemoglobin beta gene on chromosome number 11. Okay, so here let's see, in case of this hemoglobin. So hemoglobin has two parts, heme. So this is the heme part, which means it's the iron part, and the globin is a protein part. So globin is a protein part, and this protein part is made up of four polypeptide chains. Is made up of four polypeptide chains. Okay, so here these four polypeptide chains, okay, out of these four polypeptide chains, two are, okay, alpha polypeptide chains, and the remaining two are beta polypeptide chains. So out of four polypeptide chains, two are alpha polypeptide and two are beta polypeptide chain. So it is in this beta polypeptide chain, okay, there is a change in an, okay, amino acid. Okay, so let's see this one, how there is a change. So in case of normal gene, so we'll take it as a HbA. So I'm going to take as a HbA. HbA is a normal gene, and HbS will be the sickle cell. Okay, is sickle cell anemia. For that, I'm going to take a HbS. Okay, so normal gene is HbA, and sickle cell is HbS gene. So let's see here, the normal gene is having a sequence. So normal gene is having, that is a HbA, having sequence GAG. Okay, it is having, okay, GAG. So here I'll just mention three nitrogen bases will code for one amino acid. So earlier, while discussing mutations, so I have shown you that the gene will be read, okay, three nitrogen bases. Okay, so 3 and 3, this is the how it will be read, I told you. Okay, so that's what again I'm mentioning here. So three nitrogen bases will be coding for one amino acid. So here the normal gene, okay, the two strands of the DNA, so one is having GAG, another one the CTC. So this is the, okay, normal sequence. Now this CTC will be acting as a template strand. That part I will be discussing it later, and will help in the synthesis of mRNA. And the mRNA will have sequence GAG. So this is the sequence which is present on mRNA. So this one stands for messenger RNA. Okay, so here the CTC, so this is one strand of the DNA, this is another strand. So out of the two strands, one will act as a template strand and help in synthesis of mRNA. and this mRNA will undergo translation. Okay, transcription and translation. Don't worry if you do not understand now. I'm going to discuss those uh, very detailedly in the next chapter, molecular basis of inheritance. Okay, so here this GAG, the three nitrogen bases, as I told you, they are going to code for a particular amino acid, and that will be, okay, glutamic acid. So it's going to code for, okay, glutamic acid. So that I'll just mention it as GLU. So this GLU stands for glutamic acid, and this glutamic acid is present at sixth position in the beta globin chain. Now coming to the fifth one, so we see here there is proline, okay, and then, okay, so this is the fifth amino acid. Then the fourth one we have 309. I just mentioned PRO, it is proline, it's an amino acid. THR, this one stands for 309. This is another amino acid. And then leucine is another amino acid. Then histidine is another amino acid, and the first one is valine. So 1 2 3 4 5 6, okay, and the seventh one is also a, okay, glutamic acid. So LEU, so this is leucine. Then HIS, okay, histidine. And then VAL, this one stands for valine. So here all these are amino acids, are all amino acids. Okay, so these amino acids will be arranged in a particular sequence depending on the, okay, nitrogen base sequence. Okay, so this is what in case of a normal gene in the beta, okay, chain of hemoglobin. So this is beta chain of hemoglobin, right? Okay, so in the sixth position we have is this glutamic acid. So this one actually, okay, codes for, okay, so this one codes for, okay, glutamic acid. So that is okay, now what happens is this T is replaced with another nitrogen base, that is A, that we will see in case of sickle cell anemia. That condition is a HBS. So in this case what happens is, okay, sickle cell anemia gene we will see as G, okay, so here I told this is replaced with A, then it will become T here, G, then C, A, C. Okay, so you can see this T is replaced with the, okay, A, so one nitrogen base is changed. So hence sickle cell anemia is an example for point mutation. So it's an example for point mutation. It's an example for point mutation, right? So okay, and I'll just round it off. So you can see this one is being, okay, so thymine to adenine, there is a mutation. as a result, it produces mRNA that will have, okay, GUG, right? So here for adenine, the opposite one is thymine, but in case of RNA, we know it is uracil, so hence we have GUG, right? So on mRNA. Now if this one undergoes the process of translation, so this GUG will not code for glutamic acid. In fact, it will code for valine. So this one codes for valine. So you can see there is a change in the, okay, amino acid. Okay, so the remaining amino acids will remain same. Okay, proline, this will be fifth one, then threonine, and then leucine, then histidine, okay, and then valine. Okay, so here just no space, I'm just mentioning. Okay, right. So here this is the first amino acid. So 1 2 3 4 5 6, this remained as it is. Even here, this is the seventh one, the glutamic acid in the seventh position, also it remains same. Only, okay, at the sixth position in the beta chain, we can see there is a change in the amino acid. So here, this is what, so this is the normal condition, and this is a mutated condition. You can see the change in the amino acid. So this results, okay, in change in the whole polypeptide chain, right? So I'll just mention, so there is a change from glutamic acid to valine. So there is a change from glutamic acid to valine. So in the normal condition, we find in the sixth position of the beta chain glutamic acid. So this is a mutated beta globin chain. So now what is the problem with the, okay, a mutated beta globin chain? So what happens is this mutated beta globin chain will undergo polymerization when there is low oxygen tension. Okay, so in low oxygen conditions, what happens is this beta globin, so that is mutated beta globin chain, the mutated beta globin chain will undergo polymerization, will undergo, okay, polymerization under low oxygen tension. So all the molecules, the hemoglobin molecules, they, okay, join together. Okay, so that's what we call polymerization. And because of that, usually the shape of RBC, the shape of RBC is biconcave. So the normal shape of RBC, so the normal shape of RBC is biconcave. So this is what you will see from sidewise. Okay, so when we see it from side, it looks like this. This is the shape of RBC. It's the biconcave. The normal RBC will be biconcave. But what happens due to polymerization of hemoglobin molecules due to low oxygen, okay, the shape from this, okay, biconcave condition, it will be changed to an elongated shape in this way. So this is what we call the sickle shape, is the sickle shape, right? So it becomes a sickle or So that is the sickle shape of RBC. Due to this mutated beta globin, right? So what happens is this again, when high oxygen content is present, it again comes back to biconcave condition. Again it becomes sickle cell anemia, and in course of time, okay, it will get burst, and as a result, in course of time, the person will have less, okay, hemoglobin. So that's the reason why we have mentioned anemia. So anemia is a condition where we see presence of less hemoglobin content. Okay, right. So this is how the sickle cell anemia is caused due to a point mutation, right? So it's a point mutation that occurs in the gene that codes for beta globin, okay, polypeptide chain, right, which is present on chromosome number 11, right? So that's all about sickle cell anemia. Next we'll see about phenyl ketonuria. So the next genetic disorder we'll see about phenyl ketonuria, right? So this phenyl ketonuria is a autosomal recessive, it's a autosomal recessive genetic disorder. So it's a autosomal recessive genetic disorder already that I have mentioned, okay, earlier. Now this we also call it inborn error of metabolism. So it's a inborn error of metabolism. So here the gene which codes for phenylalanine hydroxylase. So here there is a gene, okay, that will be coding for an enzyme called phenylalanine hydroxylase. Okay, in short, I'll write as PAH is the enzyme, okay, coded by a particular gene, right? So this gene is present on chromosome number, so it is located on chromosome number 12, okay, in case of human beings. So on our chromosome number 12 we have this gene that codes for this phenylalanine hydroxylase. Now what this enzyme does, so here the enzyme actually converts an amino acid called phenylalanine. So phenylalanine is a amino acid, and it's going to convert this phenylalanine into another amino acid called, okay, tyrosine, into tyrosine. This is amino acid. Okay, so the PAH is going to convert, the pH is going to convert the amino acid, okay, phenylalanine into another amino acid, tyrosine. Okay, so now what happens if the gene undergoes mutation? Okay, so here the mutated gene, the mutated gene will not be producing this enzyme, okay, no enzyme. So that is PAH is not produced. The phenylalanine hydroxylase enzyme is not produced when that gene undergoes mutation. As a result, what happens is, okay, the phenylalanine, so this is amino acid, this remains as phenylalanine, okay. right? So that is no PAH. In the absence of enzyme, the phenylalanine remains as phenylalanine. And now what happens in this condition? It starts, okay, accumulating in the tissues. It accumulates in the body tissues, and okay, this will be actually converted to phenyl pyruvic acid and its derivatives. So this phenylalanine, okay, it gets accumulated and forms into phenyl pyruvic acid plus derivatives. So it forms okay, its derivatives, right? So phenyl pyruvic acid plus derivatives are formed from this phenylalanine when there is no enzyme phenylalanine hydroxylase, right? And accumulation of this, so accumulation of phenyl pyruvic acid plus, okay, its derivatives will result in, so it results in, okay, certain conditions. So there will be mental retardation. So here this accumulation, so this is due to, okay, due to accumulation in brain. So the accumulation of that phenyl pyruvic acid and its derivatives in brain will result in mental retardation, okay, and then even, okay, reduction in skin and hair pigmentation. So there will be reduction in, okay, skin and hair pigmentation, and also we see that this phenyl pyruvic acid and its derivatives have a poor absorption in the kidneys. So hence these are excreted through urine. So these are excreted through urine. Okay, so we have already seen this phenyl ketonuria as an example for pleiotropy. Okay, so one gene showing multiple phenotypic expressions. Okay, right. So with this, I would like to end this session. So we'll see the remaining Mendelian disorders in the next session. Okay, so after that we'll see about chromosomal disorders.